Hello, guys. Welcome to BIOC 2023, Day 2. For those who don't know me, I'm Wes Wilson. I'm a tumor immunologist and computational biologist at Penn Medicine Cancer Center. I have the privilege to introduce the Cancer and Evolution session for you this morning. If you have any questions for the speaker, you can put them into the Q&A and hop in, and Tim will find them for you later, or we can take questions at the end. It's going to be 9 minutes per speaker with 1 minute of questions. If speakers go over their time, me and Tim will maybe stand up, and if you keep going, we'll just get uncomfortably close to you, like on a subway, and just like get in your personal space until you leave the podium. For those of you in the room, please remember that there are microphones active on the ceilings here. People online will be able to hear your snide comments or discussing how bad our breakfast was, or there wasn't enough coffee, so keep that to a minimum. With that, I will introduce our first speaker. We have, and by the way, I'm terrible with names. Every name's going to get butchered. So the first speaker is Sarah Stankiewicz. Not even close, actually, but you're too kind. You're very kind. She's a bioinformatician at the Brigham and Women's Hospital. Her talk is OntoProc, Ontology Inferences for Bioconductor. And I'm just going to share your slides for the live people before we start. Your clock. Boom. You can use this mousey right here.
Hello, everybody. Good morning. Welcome to Boston. I know you've been here for a day, but it's a fun place. I'm going to be presenting a package that we have on both GitHub and, of course, in Bioconductor, OntoProc. Both me and Vincent Carey work on this. It's the hold on. Yes. So it's an ontology package. So what is an ontology? So for those who don't know or just need a little recap because we haven't had enough coffee, an ontology is a set of concepts and categories in a subject area or domain that shows their properties and relations between them. So two examples of one of the big ontologies that get used a lot in this package is the cell ontology, and that is a structured, controlled vocabulary for cell types in animals. And it is not organism specific and goes from everywhere between prokaryotes to mammals. However, it does exclude plants, which have their own one. So don't worry, plants have their own one, P1. And then the gene ontology, which is one of the world's largest sources of information on the functions of genes. The knowledge is both human readable and machine readable, and is the foundation of for computer computational analysis of large scale molecular biology and genetic experiments in biomedical research. So the reason for this package, the reason is we use precise terminology to improve the communication and consistency, which is really important with the consistency, among all these diverse projects. OntoProc has many ontologies with a wide range from gene to anatomy. These ontologies are in the instances of ontology index, so that the ontologies can be easily queried and potentially utilized together, all in R. Moreover, OntoProc takes advantage of ontology X packages in R, like ontology index and ontology plot. So mainly, we went by the FAIR principle rules when we were designing this package and making it, and continually updating it. So the FAIR principles are findability. So the metadata and the data should be easy to find and easy to access for both humans and our AI technology computers. Accessibility, users need to know how the data can be accessed, possibly including authentication and authorization. Interpretability, the data needs to be interpretable, while applications for workflow analysis, storage, and processing, and it needs to be reusable so that we can, once we've used the OntoProc and we found all these findings, we can use it again to further scientific knowledge. So a basic description of OntoProc is the goal of it is to strengthen development of compelling use cases for richer interfaces for emerging ontologies. OntoProc does this by supporting and harvesting a diverse bioinformatic ontologies, making particular use of, again, ontology index package on CRAN. We provide snapshots of key ontologies for terms about cells, cell lines, chemical compounds, anatomy, and to help analyze genome scale experiments, particularly cell compound screens. And just so you know, because I'm the one that does this, we update them about every 6 months. So they're usually pretty up to date. So now into some examples of functions that we have in OntoProc. So we got OntoPlot2. This function takes a list of ontology terms from whatever ontology you want to use. In this case, we're using CellOnto, and it makes a hierarchical graph with the given terms you've given it. And it runs quite quickly. So it's really nice. This is another example which we use GeneOnto instead of CellOnto to give us these hierarchical graphs. Then we have sim to CellOnto. This function you have to use cell ontology, Pronto ontology to identify cell types, defining conditions in which a given gene is named. The function returns this data frame next to it. If any hits are found, the field condition abbreviates the identifiable parts of the cell, whether it lacks a plasma membrane, has a plasma membrane, or part. Another example using cell line and then we have CTMarks. CTMarks is a Shiny app function that takes in cell ontology from GetOnto. The application plots hierarchical graphs for any cell ontology class for a given table related to tags. So this one is a Shiny app, so it has 3 parts. So it gives us this hierarchical graph that we had before on its first page. Then on the tags, it will give us the table generating the information about the query and the type of cells. And the about is, again, more tables and information about the data that you're currently using. It is very helpful and nice, and especially easier for our non-coding counterparts to use and to utilize. And then this one is the last thing is find common ancestors. This is a little bit wordy, but I'll try to get through it quickly. This function identifies all the terms in G that are the latest common ancestor of any subset terms in the character vector. And LAC is one that has no children that have the exact same set of descendants in terms of character vectors. i.e. it's the most specific term for that set of ordered descendants, knowing that LAC is useful for deciding how terms should be rolled up into broader definitions in downstream applications. Usually when the exact terms in a character vector are too specific for practical use. The descendants data frame in each row of the output describes the descendants for each LAC stratified by their presence or absence in each entry of the character vectors. This is particularly useful for seeing how different sets of terms would be aggregated into broader terms, e.g. when harmonizing annotation from different data sets or studies. Note that any names for will be reflected in the columns and data frames of LAC. Here's an example. It's mostly a true-false statement. And here are the current available ontologies. You will notice that many of them repeat, but if they're repeated, it's usually because it's an updated version. As you can see, I started adding the dates in the name so that you could know when exactly I downloaded it. And on top of that, you have the date that it was added into the R database. You can actually get this. I forgot to write it on here. I wrote a new function that will hopefully be coming out. So it's not in the package yet, called Get Available, that will give you this table, this exact table that I'm showing you, and show you all the different ontologies that you can use. And it continues on because again, I update this every 6 months. So where to find OntoProc? Of course, it is on Bioconductor, so you can easily go on to use Biomanager and install it. It is also on GitHub, where you can post any questions or bugs. And I would like to thank my collaborator, Vince, of course, Vince Carey, and Aaron Lung for helping. So is there any questions? Yes.
Thank you. So I use ontologies as much as possible. I think they're really, really helpful in a variety of cases, especially when trying to do large scale meta-analyses. I do a lot of work with single cell data and trying to compile single cell data sets from very different sources, and harmonizing cell type annotations is a nightmare. Currently, the best way to do it is just manually or doing a data-driven approach. But what I would really like is some sort of really best-in-class NLP approach to taking the non-harmonized cell type annotations provided by the authors and harmonizing them with a ontology of choice. Are there any methods for doing this well?
Oh, I haven't, I've built some of this package, but I've actually haven't really used it in my day-to-day life. I can get back to you on that because I'll look up some other functionality and see if we have that. I know one of our biggest problem right now is these ontologies are getting huge.
Yeah.
So even though we don't have them in the package and you only download them when you asked to, it can get lots of time consuming. So that's our problem, but I'll look that up for you. Sorry about that.
Thanks.
That's the answer.
And I have another question, but I'll let other people ask their questions first.
Anybody else? I have a quick one. Oh, I'll do mine quick because I have the microphone. So when I use when using these packages, you have your data, you want to throw them into ontologies. The most time-consuming part is data wrangling, because every reference genome species lists genes as different names. Do you have anything built in that would be like, I'll just throw my row names at you and you'll tell me if the gene names are right?
I don't think so, but that is a great idea that I will have to tell Vince that we should look into something like that.
I have a suggestion.
Yes.
So I'm the author of the package OrthoGene, which was originally intended to map species names, species gene names, and species names, and harmonize them to a standardized ontology. And one of the features is it can do it within species as well. It uses the I'm blanking on the name of it now. It's a server-based database that basically will just make API calls. It'll try to align them, find all the many synonyms for gene names and align them to a common nomenclature. It's the map genes function within OrthoGene. So might be something to check out.
Sort of along the same lines, I've been working with harmonized data from the cell census resource as part of our CZI work. And even when the data is harmonized, I have bad news. It's harmonized at different levels. So walking up the tree with OntoProc has actually been extraordinarily useful, because once you get to the point of having harmonized names, then you get to the point of, oh dear, they've been annotated at slightly different levels, sort of like different gating schemes. So personally, I wanted to thank you for making this available. I stumbled across it as I was tearing my hair out, trying to figure out which level we wanted to cut the T cells and dendritic cells at. And I look forward to its evolution. And as you said, for example, I know the Stanford group has, I know one of the groups at Stanford has tried to apply an NLP solution to a mesh type of where on the cell ontology does this belong. When I've tried it in the past, it didn't work real well. It was a little bit slow. Yeah. So, but I will say that the good news is that even once you get to harmonized cell ontology terms, the fun does not end. So
I don't think the fun ever ends. For us.
exactly, more research is needed. I would ask, what do you think is the next big direction for OntoProc? Is there some way to make it more responsive than just updating every 6 months, or is there any suggestion process?
So I think the next process is to figure out how not to download these whole ontologies and use some cloud computing. Because right now, I mean, it's great, but sometimes depending on the ontology you load, it can take 2 or 3 minutes for just one of them, especially like GeneOnto. So that's probably our greatest focus, is to try to make this faster and more usable. And I honestly don't know how we're going to do it, but I'm looking into, that's why I'm taking all the cloud computing stuff to try to figure that out today. So that, and the suggestion about having to figure out if terms are in there from non-harmonized data would be the great one. Thank you, guys.
Okay, takes us to our next speaker, Jacob Kroll. Hopefully I did your name right too. He's an analyst in Ravi's lab at the University of Colorado. His talk is Mole Evo R, a web app for characterizing proteins using molecular evolution phenology.
Thank you for the introduction. So I'm going to be talking about a protein analysis web app called Molevolver. We mostly use it for characterizing protein domains across homologs. So to sort of orient what I'm going to be talking about, here's some of the state-of-the-art protein analysis web applications. So for structure and function, you have things like AlphaFold, where you put in an input sequence and you try to predict the structure. With Molevolver, it's similar, where you get predicted structure and function, but you get we rely on annotation tooling instead. So something like the second figure, where you have text labels that describe sequences of your protein. And then there's also String, which is great for showing protein interactions, and Volpes, the bottom right graph, which can show you the per residue physicochemical properties like hydrophobicity, philicity, etc. And then finally, there's homology, which is a common technique to look at how proteins have evolved, what are some common ancestors, which parts are conserved versus more adaptable. And this is a really important part of our web application. So we rely on a BLAST algorithm called Delta BLAST. The first part stands for Domain Enhanced Lookup Time Accelerated BLAST. So that just means this is going to be a domain sensitive search. And again, domains are just subunits of proteins that can be labeled usually with a structure or functional label. And just a quick overview on how this algorithm works is that you have a query sequence and you first align that against a domain database. And then you take the alignment of the domain hits and you use that as a query against a sequence database. And you get your output sequences. You could take those as your homologs, or you could realign, or you could regenerate an MSA from those homologs and continue back to the step of querying against the sequence database. So there's two things going on here. One is that you start with a very domain sensitive search by searching the CDD database. And then two, you can get these very remote hits by running multiple iterations. And before I do like a live demo for the app, I'm going to talk about one particular case that this has been applied. In our group, we focus mostly on bacterial systems. So we've looked at various bacterial proteins using the app's methods. And I'm going to be talking about internalins and Listeria. So for the Listeria genus, I've highlighted 4 different species here, the first plot. And what I've highlighted in red is that 3 of the 4 species here all have this internalin P domain, the green arrow. And this is the main virulence factor for the species Listeria monocytogenus that's highlighted in bold in the bottom figure. And so what we did is that we looked at how this domain is changed across the different species. They all have this domain, but only Listeria monocytogenus is the virulent species that's mostly conferring infection. And we found that this domain, specifically in monocytogenus, has this additional like 50 residue part of the sequence that none of the other domain homologs have. Okay, so shifting over to how the app is designed, this is a very basic overview. So the whole front end is hosted in R Shiny. And when you go to the app, you submit a job that we handle with Slurm. And then once your results are compiled, you can come back to the app and use your retrieval code to view the results, download stuff, and interact with it. And if you have any questions about the app after this talk, this is like a pretty, as far as I know, a pretty in-depth Shiny app. And I think we've sort of pushed the limitations of what Shiny is capable, in my opinion, because I think it's personally best for like dashboards and maybe not for like job submitting applications. So if you want to talk about that more, and also I don't have it in this figure, but both of these sides of the app are containerized too. Okay, so I'm going to do a live demo, but here's an overview of the workflow of the app. You can bring in proteins, plain sequences, or Interpro scan output, that's a domain annotation tool, or your BLAST homolog TSV. And from there, you can go through and look through all the plots, tables, graphs that we give you. So when you first arrive on the app, this is the home page, and we have example codes here. If you don't want to put in any data, you just want to check out the app, you can click one of these here. And if I go to the query data tab just to describe what we're looking at here, I chose a human voltage gated potassium channel. And you'll first be taken to this results page when you first retrieve a job. And this first diagram here is a plot of the domain architectures. So the first column here is PFAM, a pretty well curated domain database. And all of these homologs were, I guess, hit based for this ion transport domain, this brownish color. And you can also add other Interpro scan analyses. We mostly use PFAM and Phobius because they seem to cover a lot of the domains without too much redundancy. But if you want to look at disordered regions across your homologs, then you can do that here too. And so just to be clear, all the rows here are the homologous sequences from the Delta BLAST search. And then you can look at a network of domain architectures where the radius of the nodes is proportional to the frequency of the domain across all the homologs. And then the edge weight is the frequency of the domain architecture. So like how many domains are connected in this way across all the homologs. So something like this ion transport domain is often connected with this potassium channel domain. And then you can view the distribution of your homologous hits. So I actually input a human voltage gated potassium channel. But because how this homology search algorithm works, we got a lot of bacterial hits, and we can actually filter this out with the app and then reload all of these analyses once we've applied those filters. So it'll affect all of these plots. So if I go to this main data table here and I filter for the Can you hear me okay?
I can hear you, but we didn't know you were switching to a tab. So now that you're in a browser, not your PowerPoint, you're not sharing this to people online.
Oh, I see.
What was your thing? Oh, it was in a tab. I don't know where you were. Oh, you
I'll
opened up
try to
Edge,
find it.
didn't you?
Oh yeah, I probably went to default Edge.
Yeah.
Yeah.
Sorry. I don't even see the Edge browser thing
Yeah,
anymore.
it's okay. I don't blame you. So for those on Zoom, I was just showing the homepage of the app and a few of the main plots. And what I'm doing here is I'm just filtering so that I can only view the human homologs by selecting the potassium channel domain specifically. And then if I go to the phylogeny tab now and I increase the taxon levels, you can see that it's all eukaryotes now and split between mostly human and mice and rats. And then just a few more things. You can generate phylogenetic trees with associated MSAs. So this one's pretty simple because I've filtered out all the bacterial ones. But if I go back and remove this filter, let's see. Okay. And I regenerate the phylogenetic tree. then you can see there's a much larger distribution. After I remove that filter, you get a few bacterial proteins down there. And yeah, we have thorough help documentation, and all of those are linked here by these help buttons. You can find all throughout the app. And yeah, I'm probably going to go back to the slides and just do a few thank yous to the people that helped me design the app. So thank you to Evan Brenner and Ethan Wolf for heavy testing with the application, as well as the previous developers, Samuel Chen, Lo Sozinski, and Joe Burke for their developments on the app. And then finally, the people on the software engineering team at CU Anschutz, Faisal, Vince, and Dave, they've helped a lot with UI design, containerization, and migrating the server from a previous university. And here is the URL for the application if you want to check it out. Again, if you want to talk about more like Shiny related stuff and making data analysis web apps, then you can talk to me more anytime. And that's it. Thank you.
Questions from the room?
Thank you. I had two questions, but I'll do one at a time, obviously. So for, can you apply this to multiple species or strains that are related? When you were querying, it was just one. So could you do that instead?
Yeah, you can actually query, I think up to 500 proteins at the moment. I just showed a single as a simple example, but that's actually one of the better ways to use the app, is to provide multiple inputs.
Sure. And then just to follow up from that, when you created the phylogeny, so usually you have like some bootstrap, 100,000 something. Is there a way to control for that when you were creating the phylogeny?
The phylogeny was only across the homologs, unless I'm
Okay.
misunderstanding your question.
Okay. Yeah, I guess if they're homologs, then yeah. Okay, that's all my questions. Thank you.
Anybody else?
I just had a question about the hardware, I guess, behind the server. So you were talking a little bit about like the Slurm thing. I was just wondering if you could talk a little bit on like where you're hosting this and how are you communicating between, so like is the Slurm thing happening on the same compute node as the head node, or are those different nodes? And how are you, I guess, controlling for the possibility of like many users using the app and causing that to be unresponsive if there's maybe only a single R process running the actual interface, or these kinds of issues?
Yeah, that's a great question. And it's something I've spent a lot of time working on with the software engineering team. So to answer your first question about Slurm, how we have it organized is that this all runs on a single web server that has about 80 cores. And we run 3 separate containers, one for the app, one for a master node, and one for a worker node. So when you submit a job, we use Slurm. It's sort of overkill, mainly just to keep all your jobs asynchronous from the running application because of that single R process, like you mentioned. And we're using Shiny Server, which has a lot of limitations in its design. Like if you were to run this app with just like Shiny Run app, we had an issue where if I go and click and like do some analysis, somebody else on a different browser would get the same effects. So we're using Shiny Server and just the Docker containers for hosting Slurm, and this is all on a single web server. I hope that answers most of your questions.
Thank you again. Our last speaker is Natasha Gurvich. I don't know, hope I didn't butcher that one too much. PhD student from Boston University. Her talk is a mutational signature comprehensive analysis toolkit for the discovery, prediction, and exploration of mutational signatures. Do you need a browser tab too, or are you good? All right.
All right, thank you so much for that introduction. So I'm going to talk a little bit about the Musica TK package that myself and others in my lab have developed and continued to work on for mutational signature analysis. So various biological processes and external exposures contribute to mutations in a cancer genome. And a lot of these different processes have distinct patterns in the way that they cause mutations. So we call these mutational signatures. So it's essentially a probability distribution that shows how likely you are to see various different types of mutations because of a particular process. And so because there are multiple mutational processes contributing to the mutations you see in a cancer genome, there can be multiple different signatures in play. And for that reason, these signatures provide a record of exposure and also give some insight into cancer etiology. So Musica TK is a published R package that provides tools for these types of analyses, and there's support to use it in both the R console as well as with a user interface. So the general framework, the big bulk of it is uploading your data and then discovering or predicting signatures. So I'm going to walk through some of these major steps pretty briefly and show what it looks like in the user interface. So importing data is pretty self-explanatory, but it supports uploading your own files in various different forms or accessing TCGA data sets that you can upload directly. And so once you've input this mutation data, it creates a Musica object to hold all of that information. And then you can create mutation count tables, which is essentially a table detailing how many of different mutation types you have in each of your samples. So there's support for different schema of mutations. So whether that be if you're interested in single base substitution mutations or double base substitutions, indel, whatever that may be. So you can create these tables that details how many of these different mutation types you have. So once you have that, comes the exciting part. You can then discover signatures. So you can take these count tables that you've added to your Musica object and predict or discover new signatures with that data. So there's lots of different options for selecting a different method, how many signatures you want to create. And this will predict what signatures are present in that data, as well as exposures, which is essentially activity levels of signatures. So how many mutations can be attributed to these different types of signatures in each sample. And I'll show some example of what these look like in a moment. The other side of this is prediction. So perhaps you don't want to discover new signatures in your data, but you want to see activity levels of existing signatures. So the COSMIC database, this is a database of curated signatures. So perhaps you want to look at this database and see what are the activity levels, how present are these different signatures that are already established in my own data. So you can either look at a whole database of these signatures or detail and specify just particular specific signatures that you're interested in. And then see what are those exposure levels, how active are they. So a great part about the graphical user interface is making visualization of the results really easy and interactive and customizable. So this is an example of some output you might get if you are discovering 4 signatures in your data. So you get 4 signatures displayed, you can hover over to get more information. There's lots of ways to customize this, remove labels, change the scale, just by clicking a button and replotting. So you don't have to rerun a bunch of code. It's very easy to visualize and change and customize it to what you're interested in. There's also lots of ways to visualize these exposure values. Again, those signature activities. So different ways to present the same data essentially. So whether you want to sort them by exposure levels, maybe you want to view them as proportions so you can see relative activity levels instead. You can split them into groups by annotations like tumor type, see how things vary between different sample groups, split them up by signature, and again, see how different annotations or tumor types vary per signature. And so in the interface section of this, there's all sorts of options to specify that allow you to replot that same data in all those different ways. There's also lots of different support for downstream analyses like clustering and UMAP creation or heat maps, as well as comparing your signatures to existing Cosmic database signatures to see if there are any similarities. So that was just a brief overview of the package and some of the capabilities. I want to talk now a little bit about a use case example of another project I've been working on where I've used the Musica.tk package extensively to do my analyses. And so the goal of that project is to identify mutational profiles associated with carcinogen exposure. So I have 3 carcinogens I was interested in: EAP, NTCU, and NNK. And I wanted to see if I could figure out if there are distinct patterns of mutation associated with those carcinogens. So some collaborators took cells and exposed them in vitro to these carcinogens and then sequenced them. So I have this data of various samples exposed to these different chemicals that I can then import into Musica.tk and explore that mutation data. So using the discovery tool in Musica, I was able to find 3 signatures that are active in this data set. And then using the downstream tools, I could try to make sense of this and see what's interesting. Is there anything new going on here? So the first part of that involved comparing these predicted signatures to the existing Cosmic database of signatures. So this is an example of what the output of that tool would look like. So you can see my signatures that I generated from the tool compared against the Cosmic database. And there's a cosine similarity metrics to compare a little more numerically, as well as just compare visually. And so what I saw here in this example was that 2 of these 3 signatures seem novel. So the signature 1, it shows up twice, but signature 1 and signature 2, they both don't have really strong similarity to anything in the Cosmic database. Cosine similarity is not super great, and there's some noticeable differences. So these 2 signatures that I found seem to be like something new. And then to explore that further, I looked at these exposure values, so those signature activities, and I broke them down into different sample groups to see how they vary across controls versus these different carcinogen groups. And so what I notice here is one of those novel signatures, signature 2, this green one, is really, really present in NTCU samples to a high degree, much more than any other group. And so this is suggesting that this signature that's really active in NTCU is associated with mutational activity caused by that carcinogen exposure, and it's something new. It doesn't look like anything we've seen in the Cosmic database before. So a new takeaway that I was able to do with this Musica package. And so briefly, just to summarize, I tried to give a brief overview of this package and highlight one example of a way in which it can be used. But the big components of this is we're trying to make it as compatible as possible with various file types and different schema for mutation counting and different methods for doing the signature tree convolution. And it provides support for discovering signatures, new signatures in your data, as well as predicting signature activity levels for existing signatures. And then we also try to make this as customizable as possible for visualizing results and downstream analyses that can be tailored to the particular interest of the user. So with that, thank you so much for your attention, and I welcome any questions.
Thank you. So, thanks. Do the signatures only include SNVs, or can information about chromosomal rearrangements or structural variants also be integrated? And kind of a more general question following up to that, if so, are there ways of modeling sort of multi-level interactions between SNVs within structural variants?
Yeah, so right now it just supports single base substitution, double base substitutions, and indels. That is something that hopefully could be integrated as well.
Awesome, thanks. Perfect.
I was wondering if you could talk a little about how you're comparing the signatures. So you mentioned cosine similarity, but how do you determine if one of the ones that you're finding is, say, not similar to anything in the databases? Like, is there like a cutoff or something? Like,
yeah.
And then I guess a related maybe 2-part question is, how do you know if there's not like two signatures that are sort of a mixture of signatures? So maybe you're identifying something and you're not finding something similar, but there's a mixture of signatures that are similar. So I'm just curious if you've explored that. Like, just could you talk a little bit more about that?
Absolutely. So yeah, that is a tricky thing because there's not always a really strict rule of this is the same, this is not the same. Usually a cosine similarity of 0.9 tends to be a good threshold. And also just visually looking at distinctions. So it definitely isn't a specific science, which is challenging. And as you said about signatures being composed possibly of multiple ones, that's actually another thing I've been working on, related but separate, of trying to figure out how to algorithmically or standardize that process of looking at one signature and trying to decompose it into multiple ones, or seeing if it is a combination of multiple ones. So it is something that is also a bit of a work in progress because this process is very subjective. So it is a bit of a challenge and requires the user to look through and can't just rely on the computer to make the decision, unfortunately.
Kind of a follow-up. I noticed that both NMF and LDA are supported as mechanisms for finding signatures. How do you handle rank determination or number of signatures determination for the discovery process? And have you considered validating a putative cause of a signature, for example, in a cell line via an exposure, for example, as a means of determining whether the rank determination or cluster determination is off?
Yeah, so right now there's not any support for determining appropriate number of signatures. So that is something that needs to be inputted. So whether that be, testing out a few different values to see what signatures look like and see if you're getting signatures that look the same, perhaps that's telling you that that's too many signatures. So yeah, at the moment, there's no support for determining the appropriate number of signatures.
Are there any proposals running around in the field for objective function for rank determination? We've seen them elsewhere via cross-validation and reconstruction error, but I don't know if that's really a thing in the mutation signature community.
I'm not completely sure. There are some other tools focused specifically on the deconvolution that do have more complex algorithms implemented that do allow for that prediction or determining that number. This tool it's a little simpler in the actual deconvolution process, that it doesn't implement at the moment more complex algorithms to do that. But there are some other tools in mutational signature analyses that can do that.
I guess my underlying question is whether the algorithm is complex or simple. Is there an agreed upon way to determine how distinct a signature is, as opposed to per nation? Nathan's question is really a convolution of two, and I'm just not involved enough in the community to know the answer. Do you have any idea of whether that's a thing in the mutational signatures community?
I'm not totally sure, but it definitely a difficulty in this type of analysis is part of that, determining how to specify what is something, how to separate something or determine what's unique. So I think that is something that a lot of people are working on, but I don't believe there's much consensus at this moment about how to do that.
Cool. Very cool talk. Thank you.
Thank you again. That concludes our cancer evolution session. For those online, thank you for joining us. We thank all our speakers for making their way to Boston to share their work with us. And we'll see you all at the Birds of a Feather sessions or workshops happening soon. Thank you.